Dear students, today we will discuss about the sex linkage and expression of characters in the course Fundamental of Genetics offered during the second semester of BSc degree program. Today we will discuss all about this sex linkage and some of the X-linked characters with sex. So if we talk about the introduction part, then we know very well that in sexual reproductions, the segregation takes place as per the Mendelian principles of segregation. So in that case, what happened that the monohybrid ratio, we know very well that the F1 is segregated in 3:1 ratio, and dihybrid is segregated in 9:3:3:1 ratio, irrespective of their crosses. We know, students, must know about we may just understand that if A parent is crossed with B parent, we can say that it is a direct cross. But if A parents is used as the male, in earlier case, if A parent used as the female and B parent used as a male is crossed, it is considered as the direct cross. But if we consider the B parent as female and then A is the male, so this is just considered as the reciprocal cross. These two types of crosses are generally, but the result of reciprocal crosses is not much more significant. That's why we get the yield or other traits, other characters, other characters in insignificant manner. So we don't go for the reciprocal crosses in breeding program, but it is just there in reciprocal crosses. The results are identical and the ratio of segregation is also identical as per the Mendelian ratios. But there is some of the cases where this segregation do not take place. So there is something behind it that inhibit the expression of these characters in the identical ratio or in the identical results. So it may be due to the sex of progeny during inheritance, or overall this type of deviation from the normal segregation is referred as the sex linkage. It is the consequence of gene being located on the X chromosome. The earliest known example of this sex linkage was hemophilia. It is a hemophilia characters. It is you can say that it is a disorder generally found in male where the bloods are unable to clot. So that unable to clot, so it is just bleeding is very much high. So this is later on it was explained that it is a sex linkage characters. But later on Morgan done a lot of experiments and then finally concluded that the white eye gene of Drosophila is due to that of the sex linkage phenomena. So when we talk about the causes, the Morgan first postulated the causes, and that too supported by the bridges with various experimental evidences. That first causes was the location of a gene in X chromosome and that absence of alleles on the Y chromosomes. These two causes was revealed by the Morgan and then also supported by the bridges. We know that the sex-linked genes are observed are present on the sex chromosome. That is, in case of male, if their sex determination system is XX female and XY male, so in that case the male has XY chromosome and female has XX chromosome. So the sex G chromosome, sex-linked genes are present on this X chromosomes. The sex of offspring that depends on whether the sperm cells that fertilize the eggs carries an X or Y chromosomes. It is essentially required, otherwise the offspring will not have any type of sex-linked expression or inheritance. So that sex-linked genes, we can say here that any genes which are located on the sex chromosome and mostly found on the X chromosome, because in case of XY system, the most individuals has the maximum number of X chromosome, only a single pair in which one Y chromosomes is present. So the Thomas Morgan again discovered the sex-linked genes while they studying the eye color in fruit flies, that is Drosophila. Generally, in case of Drosophila, the wild type has dull red eye, but some white eyes are observed in this, and that is considered that was supposed to be the mutant, and it was the first discovery of sex-linked genes. So when we can see the inheritance of white eye color in Drosophila, we can see here that when red eye female is crossed with the white eye male, then in F1 all the individuals, all the fruit flies produced irrespective of their sexes were red eyes. But when the red eye female and red eye male were intermated together, then in F2 generations it was found that the all females were red eye, but the males, 50% males were red eyes and 50% was white eyes. So it was concluded with the experiment, this inheritance experiment, it was concluded that the gene involved in eye color is present on X chromosome. Subsequently Morgan repeated the experiments then he conducted experiments when he used the mutant white-eyed male and crossed with the red-eyed red-eyed female. Then in that case all F1 flies were red eyes. Therefore it can be understood here that the white eye trait is recessive. When F1 were uninterbreeded, they produced the F2 with different numbers, and the frequency do not follow the Mendelian ratios. The recessive individuals were explained that is due to the white color flies have lower viability. That's why it was recessive, the white eyes were not much more prominent. So all of the F2s here, one thing must be noted that all F2 of white-eyed were found. There are some of the sex-linked disorder we can discuss here. That first one is the color blindness, which are generally observed in male human. And this color blindness test, there are different tests like achromatopsia, in which complete color blindness were observed. That in that case only shades of gray was seen. The second test, the deuteranopia, in which there is difficulties in the differences between red, purple and green pulpers. the individuals is facing the difficulties in differentiating the red purple and the green purple. The next one is the proteinopia, in which the individuals face problem with the differentiating blue green to that of the red green. Similarly, the another test, tritanoopia, is in which the individuals face difficulties in differentiating the yellow green color to that of the blue green color. The next sex-linked disorder is the hemophilia. What I already told, that hemophilia is a disorder in which blood fails to clot. It means the bloods is excessive bleeding takes place, and this is common in case of male individuals. So we can see here the inheritance of hemophilia, then when in which the father is crossed with the carrier mother without hemophilia, then we can have a progeny of two sons. One is affected with the hemophilia and another one is without hemophilia, whereas one daughter has no hemophilia, but other daughter will be supposed to be a carrier for these characters. Carrier means that the individuals having one alleles is normal condition and one is the damaged conditions. Though some of the characteristic features of sex-linked inheritance is that we can deal here. The frequency, first characteristic feature is the frequency of individual that showing the recessive sex-linked characters are generally higher in heterogametic condition of sexes as compared to that of the homogametic sexes. Second, the gene governing the sex-linked traits are not transmitted from the male parent directly to their male progeny, but the male parent transmit the sex-linked characters directly to their daughter. The next character, the sex-linked genes are not located on Y chromosomes. And last characteristic feature is the region. There is some region in Y chromosomes that is homologous to the region of X chromosome for their pairing. So when there are different, as per the sex determination systems, there are different sex-linked characters like X-linked characters, Z-linked characters and Y-linked characters. So X-linked character is discussed already in the eye color in Drosophila, where the appearance of white color eye was very rare, and that phenotype was associated with the particular inheritance of particular chromosomes, and that sex-linked genes are located on the X chromosome. The wild type eye color of Drosophila is dull red, but the pure line with white eye are also available. So here alleles for white color eye is recessive. It is the characteristic features. So the white eyed female would possesses 2 chromosomes and 1 Y, and that red eyed male would possesses a single chromosome. Here in this case of white eye color in Drosophila. The next is the X-linked color blindness in humans. In human, eye generally detects the three colors, that red, green and blue. So the affected woman passes the X-linked recessive traits to her sons but not to her daughter. And whereas the affected man passes the traits to his grandson through his daughter but never to his sons. Pattern of inheritance exhibited by X-linked recessive character, and sometimes it is called as the criss-cross inheritance. We can see here the result from the defect in one of the 3 genes found on X chromosome due to for this color blindness. So we can see here the red-green color blindness inheritance when we crossed with normal color vision female to that of the color blind male. So in F1 generations we have both male and females are in normal vision. But when we consider, when we make reciprocal crosses, that the color blind female is crossed with the normal color vision male, so in that case we observe the females have normal color vision but males are color blindness. The possible inheritance of color blindness alleles is the X-linked alleles are always expressed in male because male have only one X chromosomes. In male who receive the recessive X alleles, color alleles all have color blindness, but female will be only color blind when they have two X recessive alleles for color blindness. So carrier, we already told that the carrier are those individuals which have two alleles, one damaged and one regular alleles. Here we can see here the female mother is the carrier where half part is the affected and other alleles are strong or regular. We can see here the X-linked recessive characters and the carrier of the mothers, the unaffected fathers when crossed with the carrier mothers. So in the F1 generation, in the progeny, we have one son, unaffected son, one unaffected daughter, where one affected son and one carrier daughter. So next is the Z-linked characters. Generally, there are, as per the sex determination system, there are different system of sex determinations. So in this case, Z-linked characters, it was found in ZZ and ZW system of sex determination, which was generally found in Indian blue fall, where male has homogametic conditions of ZZ, whereas female are of heterogametic conditions, sexes, that is Z and W. Similar to that of the X-linked character, except that the pattern of inheritance in male and females is reversed. Since the males are homogametic condition, that's why it is reversed here. Z-linked characters is the cameo phenotype in Indian blue pea falls. We can see here the blue when blue female pea fall was crossed with the cameo male female which crossed with the cameo male. So in F1 we have the blue male and one cameo female, one blue male and one cameo female. In F2 generations we have blue male and one blue male and one cameo male and one blue female and one cameo females. So when this reciprocal process takes place, in which cameo female is crossed with the blue male, so in that case in F1 generations we have blue male and blue female all. But when they intermated and resulted in F2 generations, then we observed that half were blue males and 1/4 were blue females, then 1/4 were the cameo females. So this is about the sex-linked Z-linked sex traits. There are one more characteristic Y-linked characters that exhibit distinct pattern of inheritance and generally present only in male case, because male have the Y chromosomes. All male offspring of a male is Y-linked traits inherit the particular expression of the characters. But this is relatively little genetic information has observed for this sex-linked characters, Y-linked characters. Sex-linked characters inheritance, we concluded here that alleles on sex chromosome are inherited in a predictable patterns. Y-linked traits can be inherited only from the parental grandfather, never from the maternal grandfathers. Here it should be noted that the pattern of expression in inheritance was from the grandfather, not from the grandfather, never from the maternal grandfather. Paternal. X-linked characters also exhibit a distinct pattern of inheritance here. Next one is the sex influence test. Those traits which are influenced by a particular sex is referred as the sex influence test, and this trait is controlled by the genes present on autosomes. But the expression of individuals, expression of the characters is influenced by sex of a particular individuals. Generally, it is dominant in one sex while recessive in other sex, and it was due to the differences in cellular environment, the difference between the cellular environment of different sexes, and that is caused due to the caused by the production of sex hormones. The different hormonal environments affect the expression of particular traits in heterozygotes but not in homozygotes. Some of the main features of gene controlling these traits are these are located in the autosomes, and inheritance pattern was same as per the Mendelian principles, and but expressed differently in male and female sexes. It expressed more frequently in one sex than the other sexes, and expression is governed by the hormones produced by the respective sex. There was no differences observed between the reciprocal in F1 as well as in F2, as in case of Mendelian principles. We do not observe significantly the reciprocal difference between the progeny, between the individuals. Dominance in the heterozygous condition that depends on the sex of a particular individuals. Some of the example here we can see that in case of sheep, there is two types of horn conditions, that Dorset horn and Suffolk horn. When Dorset horn is crossed with the Suffolk horn in F1 generations, we have horned individuals, horned progeny, if that has male sex, but we observe the hornless individuals if that had the progeny is of female sexes. In F2 generation, we can differently observe the ratio of horned and hornless male and female progeny. Next example of this sex influenced characters is the presence of beard on some goats, that determined by an autosomal gene B, which is dominant in males and recessive in females. Here we can see when beardless male was crossed with the bearded female, then in F1 generations we have one bearded male and beardless females. But when it is in F2 generations, we concluded that we observed that 3/4 of males were bearded and 1/4 of females are bearded. So the same ratio, 3:1 ratio, is observed as in case of Mendelian ratio. Sex limited traits, there are some of the next one is the sex limited traits, in which some autosomal genes express the characters only when one sex has genopenetrance than the other sexes. It means that the expression of sex traits is limited to only a particular sexes, and that's why it is considered as the sex limited traits. The main feature of these traits is that the genes may be located either on the sex chromosome or in autosomes. In both conditions, the genes are present either on sex chromosome or in autosomes. This gene, in second, the genes control the expression of both primary and secondary characters. And some of the example for this sex limited traits are milk production in mammal, egg production in chicken, cock feathering traits in birds, and development of hair, mammary glands, etc. in human being. So one example here we can see here that the hen feathering results from a single gene H, and cock feathering result from the its recessive alleles. The expression of this H gene depends upon the presence of 6 hormones. The H gene produces hen feathering, recessive gene of H produces hen feathering if female hormone is present. Cock feathering, if female hormone is absent, we observe the cock feathering. This was proved by removing the ovaries in female or testes in male birds, which possesses hen feather results in production of cock feathering, even though H alleles, the dominant alleles of H is present here. We can see here the example, the sex-linked inheritance, plumage pattern in poultry, it is same, the sex-limited inheritance. The next example here you can see here. The differences we can see, what is the difference between sex-linked characters and sex-limited characters. In case of sex-linked characters, the genes are located on the X chromosome, but in case of sex-limited characters, the genes are present either on the sex chromosome or on the autosomes. In case of sex-linked characters, it can express in both the sexes, but here in contrast, the sex-limited characters can express only in one sexes. Next difference is it includes the sex-linked character includes the characters not related with the sex, but in case of sex-limited characters, it includes primary and secondary sex characters. The example of sex-linked characters already we discussed, that white eye color in Drosophila, hemophilia, and color blindness in men. In case of sex-limited characters, the milk production, egg production is limited by the female sex only. In addition to these sex-linked or sex-limited or sex-influenced characters, there are some of the characters which are influenced by the environment. We can see here the winged growth is influenced by the winged length is influenced by the environment, and that too is the sensitive by the temperature. In this case, an allele whose product is functional only at certain temperatures. We can see here the graph, the growth of wing is more at a higher temperature as compared to the lower temperatures in Drosophila. So another example of this environmental influenced characters is the rabbits, where the Himalayan alleles is temperature dependent, and when the rabbit is reared at the 30-degree temperature, it expresses their white color, when it reared at 20 degrees centigrade or less, then the different phenotype have been observed. So this all about the sex linkage and different characters influenced, limited or linked with the sexes. Thank you very much.